So as indicated, the scientific committee met this morning, and we had a really great conversation, a lot of good discussion. There were a few things we couldn't align on, but I don't think anyone's surprised at that. First, what did we align on? Well, one was that the importance that medications are administered with the guidance of veterinarians under a valid veterinary client-patient relationship and with a prescription. And we would really encourage all of the jurisdictions to adopt and enforce that type of regulation, because we think that would help a lot in controlling the use of the medications and understanding how those medications are being used in the racehorse. Two, we did agree that there are significant differences between thoroughbreds and standardbreds, quarter horses, and just in the basis of how they're trained, how they're raced, how the business works. I mean, that's just reality. We do not, however, find any evidence that there is a physiological difference. So standardbreds don't clear clenbuterol faster or differently than thoroughbreds do. So in some respects, I think what the scientific committee aligned on is the issue of whether it's appropriate to administer clenbuterol closer than 7 days out, the current regulation by RCI, is really a regulatory decision. It's not a scientific decision. Tom's shaking his head. 14 days, apology.
So, I wouldn't shake my head.
Thanks for keeping me straight. 14 days. So if you want to, I think we also all aligned that we thought that the risk to the health and welfare of the horse with an administration of clenbuterol, several doses immediately after racing, was minimal. Now, there's not a ton of evidence to support that. We would love more data, but the best that we could align on is that type of dosing strategy as proposed probably presents minimal risk to the health and welfare of the horse. What there was a lot of concern on, though, is especially given today's environment where we are right today, do we want to be changing the regulations for allowing a medication that has the potential to alter the outcome of a race or a horse's athletic performance? We're not saying it's performance enhancing, we're not saying it's definitely negative, but there certainly is the potential. And so that's, again, back to more of a regulatory question. Is that problematic to be altering and allowing this medication change? I think for us, the answer is, you know, it's a regulatory question.
Thank you for having me here. I'm coming here from New Jersey, where the horse is a state animal and where we have more horses per square mile than any other state. We're a small state, so you have to do the calculation there, but we also have the best standardbred racing in the country, and we have an equine science center that our motto is better horse care through science and research. So they asked me to speak about clenbuterol because my student, Charlie Kearns, you saw one of his papers featured here earlier, looked at clenbuterol quite extensively. It was a project that was funded by the Equine Science Center, not by the drug company, so we actually ticked them off quite a bit. I used some other verbiage, but that'd get me in trouble here. It was an unbiased, independent study. Charlie had come out of the human physiology background. He was a weightlifter, and he said, oh my gosh, clenbuterol, that's the drug of choice of female bodybuilders. So his question was, if you're going to give that to the horse, what's it going to do to muscle mass? Are you going to increase muscle mass? And will that translate to an increase in performance? So he went about validating his studies. He actually, if you go through the list, there's probably about a dozen papers related to his research. Very carefully done. One of the first things he needed to do was figure out how to assess if it was a repartitioning agent. And so we used a method that the folks at Cornell had validated for use in livestock species and also horses for looking at body composition. We went out and assessed whether fat-free mass or muscle mass is related to 1 mile performance. We had a lot of help. We went through Linda Toscano's barn and assessed all of her horses. And those of you that are harness racing, she won the Hamiltonian a couple of years ago, so they weren't run-of-the-mill standardbreds. And we saw a relationship between fat-free mass and performance. But then the question is, if you increase muscle mass with clenbuterol, is that going to be a functional increase in muscle mass? So we undertook Charlie's study. He wanted to get to the top of the dosing chart, the 3.6 range, and we could not get there because of the side effects of the drug, so we got to 2.4, which is 3 times the dose that's currently used therapeutically in horses. This study he had 4 groups. He had a pure control, he had a clenbuterol group, and he had a group that got clenbuterol and was exercise trained, and then he had a pure just training group. And the rationale for having the combination of training and clenbuterol was that the training may actually offset some of the effects of clenbuterol, and we're looking at a trained population. So we proceeded with the study. The horses were dosed 5 times a week. We had 2 days off. Logistics, weekends, we didn't dose, so we were cycling on and off, so to speak. They were dosed for 8 weeks. And we did our body composition measurements every 2 weeks during the trial. Everything else, all the big muscle biopsies, the measurements of oxygen uptake, et cetera, we did before and afterwards. Now, as far as the increase in muscle mass, fat-free mass, the repartitioning effect didn't become significant in the clenbuterol and exercise trained group until 6 weeks of giving that very high dose. So that took a rather long time for that, a lot of clenbuterol to get that effect, which is totally different than dosing horses after the race for the short amount of time that we want to propose for standardbreds. The other effects of the drug surprised us. We actually saw a decrease in aerobic capacity, and we saw a decrease in the ability to tolerate high-intensity exercise in our simulated race tests. So we actually had a decrease in performance with the long-term administration of a rather high dose now of clenbuterol. So it wasn't a performance-enhancing drug. The changes in fat-free mass were evident, but we weren't having a functional muscle mass that could aid a standardbred horse, which is a middle-distance runner. When we looked at the actual muscle fiber type profile, if you get in there, there are 3 types of muscle cells you have. You have type 1 fibers, which are your endurance fibers or your postural fibers. And then on the other end, you have type 2X or type 2B fibers, depending on how you classify them. And those are your sprint or weightlifting fibers. And in the middle, there's a fiber, the 2A fiber, that can sort of train either way, aerobically or anaerobically. And what we saw with the fiber type profile was the muscles got bigger, and it shifted towards what we call a weightlifter's profile, which is not conducive to doing a 1 mile standardbred race. So it sort of explains a little bit of, with the breed differences, of what goes on in quarter horses. They're a sprint animal that's been bred that way, and if you sample quarter horses without drugs, you'll see that their 2A and 2X fibers predominate what goes on in their muscles. So we were seeing the shift in the fiber area towards that ballistic type activity, which explained why we had a reduced aerobic capacity in those horses. So the bottom line was, with chronic administration, clenbuterol actually decreased performance, even though it was a repartitioning agent. The acute studies that are out in the literature, and there was one that sort of led to the rationale for a study, showed that 1 dose of clenbuterol, this was a study done in Australia by Ruben Rose and his crew, it didn't affect performance in, I believe they were using thoroughbreds in that study. It was done quite a few years ago. So the bottom line is that short-term administration of the 0.8 medically justified dose of clenbuterol is not going to enhance performance. The repartitioning effects takes much longer time. And we recommended that the 96-hour rule with the FDA-approved clenbuterol prescribed by a veterinarian, it's given, of course, after a race. And the threshold, we were bouncing that around, and the conversation went in the direction of the different states and what they have, either 25 or 20, and we recommended following what other states are doing.
Dr. McKeever was presenting the a summary kind of the minority report, because there wasn't a consensus among
I
the
think it was
commission to go to 2025. I think there was.
We were
Yeah.
kicking that around.
I was gonna.
What was the vote
I was
on
gonna.
that?
So I think that a number of the pharmacologists, let me not label, there were strong of us that felt that 25 was too permissive, that it would allow the last dose of administration of clenbuterol to be as close as 36 hours before a race. Because of the variability in how the horses clear it, those are based on, primarily on the Heather Kninch study, but I think it was also consistent with the other studies that's been reported. So, you know, I think we need to reevaluate. We'd be happy to sit down and really look at the data, the raw data of the elimination, to say if the regulators feel a 96-hour withdrawal period is appropriate, let's come up with a number. But I think most of us felt 25 was not going to be it. It was too high.
And the 20, because that was bounced around also.
I mean, the number that we were bouncing around was more like 5. So somewhere between 25 and 5, right? Like, there's a lot of variation, but I think our concern is we want to be able to say that these horses are not being administered this drug closer to race time than what the regulation is intending. And so we certainly know people can do that way and be below 25, but other nefarious folks could take advantage of that and administer a lower dose closer in, which we didn't think people wanted to be permitted.
And a point of clarification, that 25 and 20 came up because those are the rules and existing rules in states that are out there.
Yeah, and we are cognizant that that's part of the issue, is those things are already in existence. But if you're using 25, you are likely allowing administration closer than what you're saying. It's not a 96-hour withdrawal.
Mr. Chairman, just also, there was a proposal by Dr. Malin, and there was a lot of discussion about not denying the medical use of this drug. But there was a concern of its potential overuse. And Dr. Malin had suggested that in addition to making sure that there was a documented veterinary diagnosis, that perhaps the system for the reporting of corticosteroid injections in New York might be duplicated for a way for horses being identified to have a medical need be reported to the commission through a similar system. And Doc, if I'm not saying that properly, please step up and say it. But that was one of the points that was made in the committee. There was a real concern that the purpose for the administration of the medication was tied to a very specific diagnosis and was not intended to be a routine treatment, which, after hearing Dr. McKeever talk about it depressing potentially performance, I don't know who the hell would want to do that as a routine treatment, but you know, so I just wanted to put that on, just make that point.
Well, the key there to that question is, and the company backed down and redesignated how they were saying the drug should be used. They agreed that it should be at the lower dose and for a shorter period as needed.
Dr. Malin brought up the fact that there was a stable that he was aware of that the trainer gave every single horse in the stable clenbuterol regardless of a therapeutic need or not. So it's obviously, I don't want to use the word abuse, but the word abuse does come to mind. Again, open up to questions or comments from our committee first.
Mr. Gardner.
Okay, Charlie.
Dr. McKeever, could it be possible, with your research as a background, that because quarter horses are not exercised or trained as hard, that they get a much more beneficial performance enhancing effect than does the standardbred or the thoroughbred.
Well,
It's been told to us that in Louisiana that muscle mass and it's a different animal and it's built differently, et cetera, et cetera, et cetera. But this is a first for me that you said you find out that you get a better effect on performance with less training. And a quarter horse is definitely less trained.
Potentially, if you're augmenting those fiber types. However, we looked at standardbreds. As far as the effects on performance, there's the study that Rick Burks did at UPenn. It didn't look at all the markers of performance, and it certainly didn't look at, you know, the generation of power like you'd see in a quarter horse. So as far as studies that would answer that question, those need to be done. I could speculate, but then I'll get myself in trouble, and I don't want to do that.
I'm wondering if the committee looked at the potential for intratracheal use with a fallover for detection on race day, post-race samples, and was there any discussion as well as an inhaler, or did you just look at straight oral use?
The kinetic data isn't available, that we didn't specifically review intratracheal, but I think that's one of our concerns that rolls into why we think that 25 is too high, and I think that is another thing that would need to be considered.
I'll bring you the kinetic data right now. Okay?
Dr. Scalay, question.
I'm sorry.
Sure, so I guess my question is, there currently is withdrawal guidance and a regulatory threshold that is permissive of use of clenbuterol up to 48 hours prior to a race. And as an expectorant, I would think that would be a useful tool in clearing a horse's airway. I guess I'm wondering why that's not a consideration. You know, when we talk about particulate matter getting down into the horse's airway, you know, you gotta be less than 5 microns to get down deep into the lungs, so that the stuff that we're talking about is really pretty much confined to the upper airway, trachea and the large bronchi. Is that fair?
So I think guaifenesin is a reasonable alternative, but I think some veterinarians feel that clenbuterol is better at inducing enhanced mucociliary clearance. And the question that we were posed was, you know, A, does it pose a threat to the integrity of racing if this was allowed? And that's a question for the regulatories, for them. Does it pose a significant health and welfare risk to the horse? And nobody in the room felt that at the doses prescribed, it did. And so that was the other question that we had. If regulators decide that that's appropriate, I think we can come up with a level that would allow that, but that would not allow administration closer, but it's not the level that's currently proposed. So I think we're saying we're ready, we could have discussion, and I can't do this now, Tom.
I'm
Yeah,
sorry.
there's a way to
It
submit.
did not come up at the discussion. The question was not asked, but I specifically prepared an answer to that question.
Thank you.
Let me tell you briefly, I don't have it in front of me right now.
Doctor,
but-
would you mind coming up to the
Sorry.
microphone? Thank you.
If you give me my paper, I refresh my memory. We addressed the intratracheal question. We administered 90 micrograms intratracheally. Popped it into the trachea. The question I had was, when should I take my first sample? In other words, how long will it take it to hit the blood at a detectable concentration? 230 picograms per ml was the average at 10 minutes, and the concentration was dropped.
I don't really want to look at it.
Dr. Matt, there's a point of order, Mr. Chairman. There's a process by which this information should come in and get properly circulated to the Science Advisory Committee and to the members of the DTSP Committee. We did hear Mr. Williams indicate that we need to be very clear on any data that we need to further assess this, and I would ask either the members of the committee or the members of the Science Advisory Committee. I know that there were some data that was discussed. I think if somebody could email me exactly what it is you feel you need to receive, then we will then make those requests on behalf of the group. But whatever comes in gets circulated, you know, to the group.
Yeah.
And, you know, you tell me whether it's public or not public, and
It
we'll
was-
honor that.
It was published 20 years ago,
Fine.
and it's in the literature.
Thank you, Dr. Coe.
Thank you.
But I know there were discussions about access to raw data, and I just want to make sure that when I make a request to whomever I make the request to, that I'm asking for the right stuff.
Yes, Rick?
I guess this question would be to either Dr. McKeever, possibly Mary, because she was, Mary Robinson, because she was a co-author on the Pennsylvania study. You weren't? Okay.
Not a co-author.
All right. Well, anyway, in that particular study, they actually showed at the standard 8 micrograms per kilogram dose an effect measurable in their particular method of measuring percentage of muscle fat within just days. The other issue with that is that what we're talking about here is, it depends on how you actually look at it. If a horse is treated after its race and then has a 3-day withdrawal time, you're talking about a horse having either 150 or 200 days of clenbuterol a year, and I'm just a little bit confused as to how you can make a conclusion that that wouldn't necessarily have a partitioning effect. And lastly, and just editorial, we've had no clenbuterol in California for a number of years with our standardbred horses, and nobody has ever said a peep as to that being a problem.
Dr. Stanley?
Yeah, I just wanted to readdress some of the things that Dr. Cole was saying before. The reason that the majority of the scientific advisory group rejected that position at 25 picograms was because, as she stated, the concern that it could be used well within the time period of clenbuterol acceptance, too close to the race. The data that is published and been vetted in peer-reviewed journal clearly shows that you could give treatment up to somewhere between 36 and 48 hours of competition and not violate that level of 25 picograms. So it was unacceptable to the majority of the committee because of the concerns of that. There were some other members that felt, well, we have been using it with 96 hours and those levels, but there was no one that could definitively state that it couldn't be used closer. That's why it was rejected as a concept, and the level that was proposed or discussed was somewhere closer to 1 or 2 picograms as a regulatory limit that would be more consistent with 96 hours. But the data's not there to say, absolutely, this is a level that we could have with a high degree of confidence that would work for 96 hours. So we felt it was imperative to have more information to make a definitive answer for a 96-hour plasma threshold for clenbuterol in that case. And that without doing that, the high potential, as Dr. Arthur just said, for the drug to be abused as a repartitioning agent was there because it did not definitively say they could only use it 1 week and then not for another week. So therefore, they could be on about 50% of the time of clenbuterol and withdraw to compete. And those repeated doses may potentially provide that repartitioning effect. So because of the potential and the lack of being able to support the threshold, that's why the majority of the committee shot that idea down.
Thank you.
So to come back to your comments about the Nolan Watson article, that article only showed a decrease in fat mass, not an increase in muscle mass. They only measured rump fat thickness, and unfortunately, they didn't weigh almost all of their horses. They only had a scale for some of them. The design of the study, they had some thoroughbreds, I believe 8 thoroughbreds, but the majority of the horses in that study were polo horses at 2 different farms where they were either in training or sort of coming back into training. So the representative population was different than what we're talking about. But nevertheless, they only measured the change in rump fat thickness and calculated what they expected as far as fat mass. The anabolic effect is an increase in muscle mass, and they didn't report that. Also, that there are a number of other problems with that article. It was actually rejected the first time it was submitted, and it got sent elsewhere. As far as the discussion with the thresholds and
I'm talking about the article that was published in the American Journal of Veterinary Research.
Right.
A prestigious journal.
right,
Okay, thank you.
it was rejected the first time by the American Journal of Veterinary Internal Medicine, and it was based upon the review of one reviewer that's standing here. And I can give you the review that points out a whole 2.5 pages of flaws in the study. But that's a whole nother discussion.
If I may.
Dr. Robinson?
So I'm not an author on that publication, and I do understand it was rejected the first time around. However, the first author on that publication is a veterinary student, and it was her first ever attempt at writing a manuscript.
It was a good first attempt.
So, and I believe all of the valid points that you'd raised in your review were addressed, and that's why it was accepted subsequently.
No, they weren't. I read the article the second time. Body weights weren't added for all the horses, and those calculations weren't done that would show the repartitioning effect.
So despite those differences in opinion as to how complete the study is, I think the raw data that Dr. Arthur showed earlier speak for themselves, that we're clearly seeing a decrease compared to the placebo group in the rump fat.
In rump fat, but rump fat is not what's propelling that horse. It's-
Agreed. So, but as you know, I believe it was Dr. Arthur also showed, the difference between the muscle and fat. If you're losing the fat, the assumption, and you're not changing the body weight, which they did measure in the majority of the horses.
No, they measured it
I'm
in
gonna
eight of the
interrupt.
horses.
Yeah.
Interesting discussion back and forth, but we have a limited amount of time.
I understand.
And I think we've already heard the recommendations of the Scientific Advisory Committee
For clenbuterol,
for
yes.
clenbuterol.
Yes.
Mr. Chairman,
Thank you.
if I'm going to give a layman's summary of what occurred at that meeting, I think the committee was open to a decision to make it a 96-hour medication, but in a very controlled, reportable disclosure, tied to a diagnosis, reported to the commission, maintained in a database to basically guard against the routine and overuse, misuse of the drug. For a specific diagnosis, it's in the veterinary arsenal. If it's helpful to the health of the horse, I think the committee was open to that. The committee has a problem if the regulatory decision is to allow that, then the committee was certainly prepared to start assessing and analyzing data to come up with what would be a fair and appropriate threshold tied to 96 hours. There was a difference in the committee as to whether the existing published data that would indicate a 1 or a 2 picogram would be the prevailing thing. There was a difference on that. That's a decision that I don't know if the organization wants this to be further reviewed, then we would obviously need to request some data and supporting documentation to basically get to a 96-hour withdrawal time, if that's what the regulatory decision is to be. And
Which would indicate a 20 pico threshold, but I do remember saying that we need more data to support that, because there wasn't a complete consensus. Correct?
I think there's always need for more data because we're scientists. But I think reviewing the data that we currently have, maybe as a subcommittee to come up with an agreed upon, we can live with this is possible. I think it's possible to come to a consensus on that.
I think Dr. Finger was first, right?
I'm gonna pass after all that.
Yeah, because we've still got to get betamethasone and other things discussed.
So you're not going to seek my former student?
This is not an attempt at a rhetorical question. I just need clarification. In one presentation, it was stated that clenbuterol could affect the outcome of a race. In the other one, it was stated that it has no performance-enhancing effect. And I don't understand how both of those statements could be true.
I don't think Dr. Arthur said it could affect the performance of a race. He quoted the Berg study, which showed that there was no effect on performance, which is consistent with what I presented, that if anything, it goes the other way.
Mr. Chairman.
I didn't mean Dr. Arthur, I meant the other senator who was talking about how it's a regulatory decision because it could affect the outcome of the race.
So I think the general, and certainly anybody on the committee, if they feel differently, I think the evidence is that there is the potential of clenbuterol to alter the athletic performance of a horse. Some people feel it could be slightly positive. I think most of us agree with Dr. McKeever's work that it's actually probably negative. But either way, we don't really think that's acceptable. So we are trying to find a compromise where maybe you can have the beneficial effects of the mucociliary clearance and a very low likelihood of causing repartitioning effects. That's a little bit controversial of whether we can do that or not, I think, is the bottom line.
I think where we are with this is that committee will take this under further advisement. We're not shutting the door on this. Dr. Malin, if I may speak for you, is adamant that there should be a reporting requirement with this. So we appreciate your presentations, and as I said, we're not shutting the door on it, that this will be further discussed and try to establish a threshold level that would indicate a 96-hour, 96-hour
If the
withdrawal.
committee, if the organization wishes to go on it, that was not voted on at this committee, and that would have to be voted on in subsequent ones.
Okay.
Unless the committee wishes to vote on that.
Let's get an indication from the committee how they would like to proceed with this.
Duncan, hearing what's been said so far, there is still a lot of information that is unknown. I think it'd be premature to make a decision at this point.
Make a decision on what?
I would have a vote on that. I would let them, because we're talking about further study, as far as information is concerned, to make a reasonable decision on this, to try to develop a threshold for the 96-hour withdrawal time that just doesn't exist at this point. And I don't think it'd be appropriate for the committee to vote on something like that. I'd rather have the Scientific Advisory Committee develop their list as far as the information they're looking for and have it provided to them and leave the door open to revisit the site.
So what you're suggesting is have the scientific advisory committee come back to us with further findings?
Yes, yeah, because right now there's nothing definitive.
Is that acceptable to the rest of the committee?
Yes, I mean, I think where we are is science can't answer an ethical question, and the question is, do we believe it's appropriate to be permissive of this medication in closer proximity to a race than has previously been contemplated? So, and if it's the will of the ARCI to do that, then I think we need enough data to come up with a correct number and not a close enough, to Cindy's point.
And I would say for the record, I think the USTA would need to address the issue of the other medications that would accomplish the same thing, why that is insufficient. Let's not do it right now because we've got time issues, but I think that can be done in a written submission.
Okay.
So
scientific advisory committee respond to that.
Okay, super. So it is our contention as veterinarians and as standardbred veterinarians that, just as veterinarians in general, that intra-articular injections of corticosteroids are appropriate and therapeutic. High levels of athletic exertion are accompanied by stress, which results in inflammatory mediators that are elaborated in joints, and those need to be treated in a lot of cases. Over 53% of racehorses experience lameness during their careers, of which joint injury is a major cause. As many as 27% of thoroughbred yearlings go through the ring with pre-existing arthritis in their hocks, and other breeds suffer from similar problems. So I just want to lay out that we consider this to be appropriate therapeutic medication for racehorses. The RMTC recommended a 10 picogram threshold based on a study that was done a number of years ago. And we've asked for the data repeatedly and never gotten it, but in January they just posted on their website something kind of long. This is Dr. Sams's data, and it says, in order to ensure that intra-articular injections were performed with sufficient time. So it's basically a scientific paper, except it's not a scientific paper, it's just the results of a study that were posted on the internet by the RMTC. In order to ensure that intra-articular injections were performed with sufficient time for the veterinarian to gauge the efficacy of the treatment, the threshold was based on intra-articular data obtained at 7 days, including estimated concentrations between the LLQ, which is the limit of quantitation or how low they can conclusively say what the level is, and the LOD, or the limit of detection. So they know that it is the drug, they know that it's there, but they can't really tell you how much. So even though they can't really tell you how much, they estimated these concentrations. The data were log transformed, and the mean and the standard deviation were determined. And they calculated a threshold based on a 95-95 tolerance interval. The calculation for that tolerance interval used a K-factor for 20 horses. So they had no measurable values at 7 days. They estimated numbers, and then they performed a 95-95 statistical calculation using a K-factor for 20 horses. They got a threshold of 7.9 picograms. They rounded it to 10, and they called it good. Earlier, we got a spreadsheet that was Dr. Sams's raw data, and apparently not all of these data are correct because it lacked the appropriate qualifier ions. Maybe there was a deuterated standard that wasn't right, but there were some problems with this data. Nonetheless, this is the only data that we know that's in existence for this. If you take those data, and certainly it might not be the right data, but it's going to approximate the data, and it's certainly going to approximate what the data did. And if you take that information and you apply appropriate statistics to the data, which is not a 95-95 tolerance, what you actually get at 7 days is a threshold of 41 picograms per milliliter. So this is RMTC's own data. If you take the data at 9 days, so this is 2 days later than 7 days, because there was one outlier horse that had a high spike at 9 days, you actually get a threshold of 75 picograms per milliliter. So my point is, even the RMTC's own data argues that the threshold should be higher than 10 picograms. So just going back to, there's a lot of shenanigans associated with the RMTC data. We don't know what it is. Apparently it was put on the website without Dr. Sams's approval, or at least he doesn't say that this is actually his real data. And so we actually don't know where the RMTC threshold came from. Now let's dial it back to what the Harness Medication Committee recommended. So at the current recommendations of 100 milligrams of methylprednisolone or betamethasone for a horse at 21 days, or triamcinolone at 9 milligrams in a single joint or betamethasone at 9 milligrams in a single joint, you don't even have a single intra-articular corticosteroid that you can use in the stifle joint of a horse. What's the significance of that? The stifle joint is a very large joint compared to the ankle or knee. So what we've done is we've generated thresholds for knees or ankles with God knows what numbers and God knows what statistics, using a dose that's appropriate for those joints, and we're trying to apply it to the stifle joint. So as it is today, if I have a standardbred in my practice and the race has been deep, now we kind of all, the racetrack has been deep. Now we kind of all know right now, standing in California, that the condition of the racetrack can be fast and hard, and it can be deep, and different conditions of the track do predispose to different conditions for the horse. So a deep racetrack for a standardbred is going to result in a lot of stifle conditions, and those will usually be bilateral, meaning both the right stifle and the left stifle. And so the dose that is required of betamethasone for a stifle is 15 milligrams. If you're going to treat them both, that's 30 milligrams. And if you go to all of the previous literature, of which there's only 2 papers, Knitsch paper in 2016 and the Menendez paper a few years earlier, both of those studies, 30 milligrams is the dose that's used most commonly. So it's the practitioner dose, it's a practitioner recommended dose, it's the only dose that you can use really realistically in the stifle of a horse. Betamethasone is a safe drug compared to some of the other corticosteroids. It causes less damage, and there's no reason that a 7-day withdrawal is required for us to inject a horse and then assess response to treatment and then race the horse. There's technically no difference between 6 days and 7 days. 6 days gives us a similar amount of time to inject a horse, assess response to treatment, and race it without interfering with the weekly racing program of the standardbred horse. And so that's where our dose recommendation came from. That's where our withdrawal recommendation came from, and we feel that it doesn't pose any threat to the integrity of horse racing. One of the concerns that's been brought up by the RMTC and some of the regulators is, well, with this threshold of 100 picograms, somebody could inject a horse 3 days out, 4 days out, 3 days out. They could inject, wouldn't be the stifle in this case, but it might be a lower dose in an ankle. And so now they're too close. And so it's the same argument as the clenbuterol. Well, now they can do all kinds. Well, their current regulations already allow that. I can inject a horse with dexamethasone and get past the drug test. I can inject it with isoflupridone, and I can actually inject an ankle with depomedrol at 5 days, and still, and that's from a research paper that I did with Dr. Tobin and Dr. Malin, that we showed very clearly that you could inject an ankle with depomedrol well under 7 days and not have a positive drug test. So it's a little disingenuous to say, well, we did it with these drugs, but with this drug, we're not going to let you do that. So that was the justification for where this came from. The 100 picograms is somewhat based on a threshold they had in Pennsylvania that was effective for a long time. But actually, we also have a current study where we're currently analyzing data, and the preliminary, Dr. Malin indicated to us that the preliminary results are showing that 100 picograms would be appropriate for a 6-day stifle injection with 30 milligrams of betamethasone.
So, I mean, first, Claire, I don't think it's appropriate here to get into the weeds on all of the details at the risk of everyone falling asleep. So we discounted some of that data, but it makes a lot more sense now. I know what you're talking about, that data that was in a spreadsheet that was more than raw data. I would say it was unanalyzed data. And so we said, let's not talk about that. Most of what we were discussing was the Knitsch paper. And I think what we felt is, yes, standardbreds, again, there's some differences. Yes, we see the need that maybe you might want to inject more than one joint. But there is still that concern with a closer administration. And we really could not see where you came up with 100. So that was kind of our biggest, show us the kinetic data of where you came up with 100 at 6 days. because all of the studies that we were looking at showed that it's actually below the limit of detection at 4 days. And so, you know, that was really what we're wanting, is wanting to see the data of how you arrived at 100.
In New York, we've cut to the quick on a lot of these by making sure that the injections, whether it's betamethasone or clenbuterol, they have to be done by a veterinarian. Not a small animal practitioner from Timbuktu, but a veterinarian licensed by the specific gaming commission. That's item one. Those injections have to be documented. The amount, the type of injection, and which ankle, if you will, and it has to have a diagnosis. And the veterinarian has to inspect that horse. Becomes a veterinarian requirement. Now, Dr. Palmer can address this much better than I, but with the joint injection schedule we have in New York, it nails it down pretty good. With clenbuterol, the easy way to get away for a lot of the problems is it has to be specified as an authorized product, not a concocted one. And you have to specify it. So if that is done with many of these injectables, that'll get rid of a lot of problems. Obviously, somebody can come in close. Somebody can use pharmacopuncture, okay? Inject the acupuncture, if you will. It can reduce the dose immensely. So, but I think what we really need to cut to the quick, until data is available, is to require rigorous record keeping.
And I think everybody on the committee, that goes back to our mutual discussion, that we think that that would be really helpful. And then the other comment that I think Dr. Stanley made at the end of the meeting was the need to reevaluate these thresholds. They're not written in stone. So if they're not right, if we have more data, if something needs to be adjusted, I think we need to have an open mind to be able to address, and we may need to do some additional studies to reassess some of the kinetics on these drugs.
Not only is it true for clenbuterol and betamethasone, it's going to be critical for drugs such as the cannabinoids, et cetera, et cetera. When you get real close to the threshold, you have to have very rigorous data.
Yeah, I think we're all agreed on that.
I'm going to give you my layman's review of the Scientific Advisory Committee's meeting. It's already been said, diagnosis, documentation, and lack of data. So in order to move forward, we need to review more data. We need to require diagnosis and the documentation.
Mr. Chairman, I think, I know there was a request to Dr. Knitsch for her raw data, and she agreed to share that. I think there was a discussion about possibly a field study that Dr. Fenger may have conducted, and then a request for data on that. And Dr. Benson.
Actually, Dr. Benson, let me just say if any of the committee members have any questions or comments first. Hearing none, you're up.
Good afternoon, Dr. Dion Benson from the RMTC. Couple of comments. I'm not going to get into statistics. I think Dr. Fenger and I disagree about those. We've had a statistician or an epidemiologist look at our statistics, and I'm very satisfied with that information. But there are two things to note. The manufacturer's recommended dose for Betavet, which is the product that was examined in our study, is up to 2 joints at 9 milligrams each. That is not a number we made up. It is not something we got out of the air. It is the manufacturer's recommended maximum dose. The other point is, if you look at the 7 days, it was a recommendation that was from the AAP, the 5,000 veterinary members of the AAP who care for horses, including racehorses, and that was their recommendations, that we start with a 7-day withdrawal. The final thing I'd like to say is that there has been a clinical study. Dr. Knitsch presented at the AAP in 2016 a clinical study of numerous joints with numerous corticosteroids that was participated in with numerous practitioners in the California racing circuit. The results of those studies showed that, with the exception of doses between 30 and 60 milligrams in the stifle, those doses cleared in 10 or fewer days. So, to me, these drugs are available. If you get them in the fat pad, if you miss the joint, you're going to have problems. We all know that. And as Dr. Fenger herself said, there are other drugs she can use. So I'm not sure why we're here arguing this. Thank you.
I'm going to try to make this really quick.
Yeah,
So
I don't want to get in an argument on this. Just present facts, nothing more.
the manufacturer's recommended dose is a recommended dose by the manufacturer. That has to do with things like they have to do toxicity studies where they do 10x that dose to determine if it's toxic, 5x that dose for multiple treatments to determine if it's deleterious. So they always choose the lowest dose. There are a number of drugs, innumerable drugs, where we go to a higher or even lower dose than what they recommend. That is not a maximum recommended because that's what you have to do. There's an entire law, the AMDUCA, that tells us that we can use stuff off-label if we deem it to be clinically relevant. And that's 30 milligrams is the dose that is most common in the literature for what's used clinically as recommended.
Claire, let me help you out. The Scientific Advisory Committee admitted there's extra label drug use. It's appropriate. It's based on the veterinarian's indication of what they think they should need. So I think we need to be careful to say, 30 milligrams may be appropriate. I mean, I think it just comes down again. We need to have some meetings. We need to have some discussions to see what the data is out there, what maybe we need to do to shore it up to get more. I think the view was, as with clenbuterol, we're open to having some discussions and sharing data.
As someone who has administered the 10 picogram level for, I guess, coming on 5 years now, you have to work to get a betamethasone positive. If anything, the level is too low. And in California, no, it's too high. You're right, because horses, I review all inarticular violations. I think we've had one in standardbreds, and the entire time we've had the regulation. In thoroughbreds, they are routinely used at 72 hours, and when people get into problem is when they inject high suspensories with them. If you use betamethasone at 7 days, boy, you really missed the joint.
I'm so sorry. I don't want to get into this, really didn't want to get into this, but in California, according to the laboratory reports that I have seen, you're screening for corticosteroid in pooled urine samples. So the laboratory is taking an aliquot from one sample, an aliquot from another, urine sample, mixing them together and screening for corticosteroids. And then if it's positive in the urine, pooled sample, then they're confirming it in blood. There's not another laboratory in the country doing that. That's why you're not getting corticosteroid positives. It's got nothing to do with the threshold.
Clara, I'm not going to argue with you. It's just another example of you not knowing what you're talking about, so I'm going to leave it at
Wait a
that.
second. Let's not go there. Unless the committee, anyone on the committee has a different opinion, and please voice it, we will have the Scientific Advisory Committee continue to look at the data. We're not closing the door on your request, but we don't have enough right now to make a decision or recommendation on it.